Good afternoon, everyone. Myself Sk.Ismaiel Basha, Department of Botany, K.B.N College. Today discuss about fourth semester genetics, plant breeding, and cell biology. The first chapter, cell. Cell theory. All living organisms are made up of one or more cells. The structure, shape, and functions of living organisms depends upon the cells. Plant cells were discovered by Sleden, German scientist, and animal cells were discovered by Schwann, British scientist. The two scientists proposed this cell theory. According to it, the cell is the structural unit of all living organisms. Cell is the functional unit of all living organisms. But they could not explain how these cells are obtained. Rudolf Virchow discovered that new cells are obtained arise from pre-existing cells due to cell division. Now the cell theory was modified as below. The structure and function of living organisms are based on the cells and products of a cell. All cells arise from pre-existing cells. The difference between the prokaryotic cell and eukaryotic cell. The small and simple is a prokaryotic cell and eukaryotic cell is a large and complex. The 0.1 to 5.0 micro in size, 10 to 100 micrometer in size. Unicellular or multicellular. Nucleus is absent, but eukaryotes nucleus is present. Circular DNA present in prokaryotes and linear DNA present in the eukaryotic cell. The single haploid chromosome present and the paired diploid chromosome present. Lack membrane bound organelles present. Has membrane bound organelles. The reproduce both sexual and asexual, mostly reproduce sexually. Cell division by a binary fission and mostly reproduce sexually. The cell division by the binary fission, the cell division by the mitosis. Examples are bacteria and archaea cells. The examples are plant and animal cells including the humans. This is the eukaryotic cell and prokaryotic cells difference diagrams. The animal cells versus and plant cells. A plant cell is surrounded by the rigged cell wall. An animal cell does not have a cell wall. The presence of a large vacuole is seen in plant cells, whereas there are a very small vacuoles are compared to plant cells are seen in the animal cells. Larger in size in plant cell, the animal cells smaller in size. Plant cells have a plastids, animal cells do not have a plastids. Centrosomes are absent in plant cells, animal cells have a centrosomes. Plant cells do not have a cilia, animal cells have a cilia. Lysosomes are very rare in plant cells, the animal cells have a lysosomes. This is the pictures, the prokaryotic cell, the animal cell and plant cell. The ultrastructure of plant cell. The plant cell is rectangular and comparatively larger than animal cell. Even though plant and animal cells are eukaryotic and share a few cell organelles, plant cells are quite distinct when compared to animal cells as they perform different function. Some of these differences can clearly understood when the cells are examined under the electron microscope. The plant cells shows the cell wall, cell membrane, nucleus, plastids. The cell wall, it is a rigged layer which is composed of a polysaccharides, cellulose, pectin and hemicellulose. It is located outside the cell membrane. The cell membrane, it is the semi-permeable membrane that is present within the cell wall. It is composed of a thin layer of protein and fat. Nucleus, the nucleus is a membrane bound structure that is present only in eukaryotic cell. Plastids, they are membrane bound organelles that have their own DNA. They are necessary to store starch and to carry out the process of the photosynthesis. The ultrastructure of cell wall. The cell wall rigged semi-permeable protective layer in some cell types. Cell wall is positioned next to the cell membrane. In most plant cells, fungi, bacteria, algae and some archaea. Animal cells, however, do not have a cell wall. The cell wall conducts many important functions in a cell: protection, structure and support. The cell wall composition varies depending on the organisms. In plants, mainly of strong fibers of the carbohydrate and polymerase cellulose. The cellulose is the major component of cotton fiber and wood and is used as a paper production. There are at least 8,000 to 15,000 glucose monomers for cellulose molecules and are 0.25 to 0.5 micrometer long. The molecules are flat and ribbon-like and lie parallel to each other. Hydrogen bonds occur between the molecules, thus crystallizing and producing the aggregates. These aggregates are called microfibril. Each microfibril contains 40 to 70 chains which lie side by side. And this can be seen in an electron microscope. The space between the microfibrils are filled up with lignin, cutin, pectin substances, hemicellulose, water, etc. Thus, the microfibrils gain considered as the strength. In primary cell wall, the orientation of the microfibrils is transverse to the long axis, and during growth the arrangement may be longitudinal. The orientation in secondary wall may differ from a primary wall. Tracheids and fibers shows three layers in their secondary wall: the outer layer, the central layer, and the inner layer, among which is the central is the thickest. The S1 and S2 layers lie adjacent to primary wall and cell lumen respectively. These layers S1, S2 and S3 may be distinguished by their respective orientation of the cellular microfibrils. In S1 and S3 the microfibrils are in the form of a lax helix, and in S2 it is a tip 1. Ultrastructure of a plasma membrane. Plasma membrane is also referred to as the cell membrane. It is the membrane found all cells that separate the inner part of the cell from the exterior. A cell wall is found to be attached to the plasma membrane to its exterior in plant and bacterial cells. Plasma membrane is composed of a lipid layer which is the semi-permeable. It is responsible to regulate the transportation of materials and the movement of the substances in out of the cell. Plasma membrane function. This membrane is composed of a phospholipids bilayer implanted with the proteins. It forms a stable barrier between the two aqueous compartments which are towards the outside and inside of the cell in the plasma membrane. The eminent proteins perform the specialized function which include cell recognition and selective transport of molecules. Plasma membrane renders protection to the cell along with the providing the fixed environment within the cell. It is responsible for the performing the different function. Plasma membrane components. It is composed of the following constituents. Phospholipids form the ultimate fabric of the membrane. The peripheral proteins present on the outer or inner surface of the phospholipids bilayer but are not implanted in the hydrophobic core. Cholesterol folded between the hydrophobic trails of phospholipids membrane. Carbohydrates found to be attached to the lipids or proteins on the extracellular side of the membrane, leading to the formation of glycolipids and glycoproteins. Integral proteins found to be implanted in the phospholipids bilayer. Polymorphic cell organelles. Lysosomes are known as a polymorphic cell organelles. Actually, polymorphism is the occurrence in the several different shapes particular in reference to the species to genetic variation. Lysosomes occur in more than one form inside the cell. There are two basic types of lysosomes: primary lysosomes and secondary lysosomes. Ultrastructure of chloroplast. Chloroplasts are green colored plastid present in the plant cell that are responsible for the photosynthesis. Chloroplasts are a flat disc shaped structure measuring 2 to 10 millimeters in diameter and 1 millimeter in thickness. They have two membranes, namely the outer and inner membranes. The outer membrane permits the movement of the ions and metabolites, whereas the inner membrane is highly specialized with the transport protein. Both these membranes are phospholipid layers measuring the 6 to 8 nanometer in thickness. These two membranes are separated by a gap of 10 to 20 nanometer known as the intermembrane space. Each chloroplast has two definite regions, namely stroma and grana. The fluid present inside the chloroplast is known as the stroma. Inside the stroma there are a interconnecting flattened membrane compartment known as the stroma lamella. It is the site of the dark reaction during the photosynthesis. The stalk proteins of membrane bonded flattened disc of the chloroplast is known as the grana. Each disc-like structure of the grana is known as a thylakoid, and the space within the thylakoid is known as thylakoid space. Granum is the site of light reaction during the photosynthesis. This diagram shows the chloroplast structure. Plastid DNA. Plastid DNA exists as a protein DNA complex associated as a localized region within the plastid's inner envelope membrane, and these complexes are called plastid nucleotides. Unlike the nucleus of the eukaryotic cell, a plastid nucleotide is not surrounded by the nuclear membrane. A plastome typically contains a genome that encodes the transfer of the ribonucleic acid tRNA and ribosomal ribonucleic acids rRNAs. It also contains the protein involved in the photosynthesis and plastid gene transcription and translation. But these proteins are representative only a small function of the total protein setup necessary to build and maintain any particular type of the plastid.